Welcome back to my channel. My name is Dr. Anwar Yusuf Dunbar. This is Big Discussions 76, Science and Technology. First of all, please like this video, please share it, and please subscribe to my channel. Well, it looks like I'm back with another Coronavirus Monday offering here. It just so happens that last week I found something that I thought was worth capturing on the coronavirus. And it turns out that last night I found something on Twitter that I thought was worth capturing here on my channel. As you can see, I'm doing the voiceover format, which one of my heroes and who I now consider a mentor, BGS Ibmore, he does the voiceover format for the most part, and also one of my collaborators from the same tribe, Nicole Ali, she does the voiceover format as well. So I want to shout those two out. And I may do some more of these because this is a very efficient way to create content and to get messages out there in a quality and a expedient way. So I had to wait until the groundskeepers outside finished so that there wouldn't be the sound of leaf blowers and hedge trimmers going in the background of this offering. But in any case, I found an article on Twitter last night before I retired for the night, and this article is entitled "Johnson & Johnson's Coronavirus Vaccine Protects Monkeys, Study Finds." And this was written by Carl Zimmer on July 30th, 2020. So this article is a few weeks old. It's a few weeks old, and it's from the New York Times. And at some point on this channel, I will probably attempt to just from a high level break down the science of what vaccines do. I am not an immunologist. I am a pharmacologist and a toxicologist by training. But as me and Nicole have discussed on her panel and in the interview that we did, a lot of the techniques overlap. So you can be in one biomedical discipline and understand the experiments that were done in another discipline. So I'm going to jump in here, and it's a short enough article for me to read in a format like this. So here we go. It's the second study in a week to report promising results in monkeys for a vaccine candidate. But the real test will come with human trials that are now underway. An experimental coronavirus vaccine developed by Johnson & Johnson protected monkeys from infection in a new study. It is the second vaccine candidate to show promising results in monkeys this week. The company recently began a trial in Europe and the United States to test its vaccine in people. It is one of more than 30 human trials for coronavirus vaccines underway across the world. But until these trials are complete, which will probably take several months, the monkey data offers the best clues to whether the vaccines will work. This week has been good. Now we have two vaccines that work in monkeys, said Angela Rasmussen, a virologist at Columbia University who was not involved in the studies. It's nice to be upbeat for a change, she said, but she cautioned that the new results shouldn't be used to rush large-scale trials in humans. We just can't take shortcuts, she said. Unlike many other vaccines in development, that might require 2 injections, the Johnson & Johnson candidate shielded the monkeys with just 1 dose, according to a study published on Thursday in Nature. It's a very reassuring level of protection we saw, said Dr. Dan Barouch, I think I'm pronouncing that properly, a virologist at Beth Israel Deaconess Medical Center in Boston and a co-author of the new study. The study comes just 2 days after a similar one was published on a vaccine tested by Moderna and the National Institutes of Health. But the 2 vaccines work in very different ways. The Moderna vaccine delivers a kind of genetic material called messenger RNA into cells. The cells use the vaccine RNA to produce a protein found on the surface of the coronavirus called spike protein, which then hopefully prompts an immune response. I shot a video or 2 on the spike protein back in February or March, by the way. RNA-based vaccines are being tested for a number of diseases, but none have yet been licensed for use in people. In the Moderna study, researchers vaccinated monkeys by giving them 2 shots spaced over 4 weeks. A month later, they infected the animals with the coronavirus. In some of the vaccinated monkeys, researchers could not detect the virus in the nose or lungs. In others, the virus replicated slowly before disappearing. Moderna began phase 3 trials of its mRNA vaccine on Monday, as did Pfizer, which is testing its own mRNA vaccine. So there are multiple groups working on this. The Johnson & Johnson vaccine, in contrast, is based on a virus called AD26, which researchers have modified so that it carries the coronavirus spike protein gene. The AD26 virus can slip into human cells, but cannot replicate once inside them. Its host cell then uses the spike gene to make the coronavirus proteins. This month, European regulators approved Johnson & Johnson's AD26 vaccine for Ebola. It was the first time this kind of virus-assisted gene delivery was approved for any disease. In March, Dr. Barouch and his colleagues designed 7 variants of an AD26 vaccine for the coronavirus. They made tiny changes to the spike gene to see whether they could get cells to make more copies of the viral protein. They also tested variants that would make the spike protein more stable, which might prompt a stronger immune response. Based on earlier research, Dr. Barouch and his colleagues suspected that the AD26 vaccine would be very potent. They decided to run their experiment using just 1 dose to see whether that was enough to provide immunity. After a single injection of the vaccine, they waited 6 weeks and then infected the animals with the coronavirus. 6 of the 7 vaccine variants offered monkeys partial protection against the coronavirus, meaning that the virus replicated only at low levels in animals. The 7th version proved more powerful than the rest. 5 out of 6 monkeys that received it had no detectable virus at all. The sixth had only low levels in its nose. The fact that we could protect with a single shot in animal models, was quite a positive surprise to us, said Dr. Paul Stoffels, the chief scientific officer of Johnson & Johnson. It was this best performing vaccine that Johnson & Johnson used last week to begin its first human safety trial, so-called phase 1 trial. A so-called phase 1 trial. If it goes well, the company hopes by September to enter phase 3 trials, which test not only whether the vaccine is safe, but also whether it works. The company plans on testing both single and double doses. Dr. Rasmussen said that a vaccine that proved effective with a single dose would make it far easier to treat the billions of people who need it. Theoretically, you would need less of it, so you give it to more people more quickly, she said. Inovio, a company developing a DNA-based vaccine, announced Thursday that monkeys challenged 4 months after vaccination had a reduced load of the virus in their nose and lungs. Their report has not yet been published in a scientific journal. AstraZeneca and the University of Oxford have developed a vaccine based on yet another type of modified virus called ChAdOx1. In May, they released promising monkey data, which was also published on Thursday in Nature. The team is now running phase 3 trials in people, which could produce results by early October. It's exciting to see the number of platforms that are showing promise for a vaccine, said Stacy L. Schultz-Cherry, a virologist at St. Jude Children's Research Hospital in Memphis, who was not involved in any of the trials. So that's the article. I'm going to leave this article in the description box below in case anyone wants to read it themselves. So it looks like multiple groups are working on this, which ideally is what you want. You want multiple scientific minds chipping away at the same scientific question. They can create data and they can fill in multiple pieces of the puzzle for each other. And with these multiple minds and multiple groups all pushing for the same thing, that might get us to a vaccine much quicker than we would get one if only one group was working on this. So this article, as you can see, this requires a lot of immunology and virology when you're approaching it from the vaccine side, and when you're approaching it from the decreasing inflammation and decreasing viral replication, you're approaching that more from the pharmacology side. So I'm going to stop this here. Please let me know if you have any questions or comments in the comment section below. There are some things that I heard Nicole talk about. There are some things that were covered in this article that I heard Nicole talk about in her content. So these things will probably come up when I go up on her panels, and when I have discussions with her in BGS and others. So please like this video, please share it, and please subscribe to my channel. And as always, remember that your attitude determines your altitude. Take care, and I will talk to you the next time. Bye-bye.